This video abstract is AI generated. How does Alkbh5 activate oxygen only when the correct substrate is bound? Alkbh5 is an RNA demethylase that removes M6 from RNA. Like other Alkb homologs, it is an iron 2/alpha-ketoglutarate dependent enzyme, and it carries out demethylation through two coupled reactions. First, the iron 2/alpha-ketoglutarate center activates O2 to generate the reactive oxyferryl intermediate. Second, that activated species oxidizes the methyl group on M6A, producing demethylated adenosine. This raises two major mechanistic questions. Why do iron 2/alpha-ketoglutarate and M6 bind sequentially rather than all at once? And why does O2 bind to iron 2 only after M6 is present? To answer the first question, we used solution and MR together with accelerated molecular dynamics. The APO enzyme is highly dynamic at the active site. When iron 2 and alpha-ketoglutarate bind together, these dynamics are reduced, and the active site becomes more ordered. In other words, iron 2/alpha-ketoglutarate binding creates the structural platform needed for specific M6 recognition. Next, NMR revealed something unexpected. When M6 binds to the Alkbh5 iron 2/alpha-ketoglutarate complex, residues near the active site split into two sets of peaks. This indicates that M6 stabilizes too slowly, exchanging active site states. Molecular dynamics explains what those two states are. They correspond to an equilibrium between two alpha-ketoglutarate conformations, an offline state and an inline state. In the offline state, alpha-ketoglutarate partially shields the metal center. In the inline state, alpha-ketoglutarate rearranges, opening the coordination environment around iron 2. That rearrangement answers the second question. The inline state makes iron 2 more accessible to O2, so M6 does not simply bind as a substrate. It shifts the alpha-ketoglutarate equilibrium toward the inline conformation, exposes the metal center, and enables oxygen activation. The final model is therefore a gated activation mechanism. Iron 2 and alpha-ketoglutarate bind first and organize the active site. Then M6 binding shifts alpha-ketoglutarate in line, increases metal accessibility, and triggers O2 activation only in the productive substrate-bound complex. To know more, read our work online.